Hello students, this recording is for Biology 1406. This is Chapter 6, Metabolism, the first subject matter for Unit 3. Alright, so we're going to run through this pretty quickly since we've already had lectures on those previously, and I'm posting this for review as you get ready for the third exam. So energy and metabolism. Metabolism, of course, deals with all of the chemical reactions in a living organism, all of the conversions of potential energy to kinetic energy and back and forth into different forms of potential energy. The power source for that metabolism comes ultimately in the form of energy from the environment, either in the form of light or high-powered inorganic molecules in chemoautotrophic pathways. And what we're looking at in this diagram right here is the flow of energy through a ecosystem. So we get light, thermal, radiative energy, heat energy, and all of that from the sun. Some fraction of that energy is absorbed and converted into stored chemical energy by producers, plants, and other autotrophic organisms. Those organisms have generally two paths energetically. They carry on with their life, reproduce, etc., and inevitably die, or pieces of them fall off and die, in the case of plants, which are sort of modular. And that organic debris from those living things is decomposed. Fungi, bacteria right here. It says worms. Worms are not decomposers. They're just detritivores that eat dead matter. But decomposers break organic dead matter into inorganic elemental constituents, and that's fungi and bacteria that are primarily responsible for that activity. And the energy that was originally captured from sunlight in that path right there is all going to be lost ultimately as heat. Another possibility for producers is that their tissues are consumed by consumers, animals that are herbivorous, either eating part or all of the plant. And eventually, again, all of the material that makes up those animals is ultimately decomposed by fungi and bacteria, and whatever final organic matter there was is completely broken apart, and all of the energy in the end, all of this energy that's captured can be accounted for if one is very careful and note that it's all released as heat. And in fact, every one of these energy conversion steps along the way right here releases some of the energy as in heat. The second law of thermodynamics says whatever energy is in plant matter that's eaten by an animal, only a fraction of that is actually converted into animal mass. Some of that energy must be lost as heat. Same thing here. Some of this energy must be lost as heat. Some of this energy must be lost as heat. So that's a little bit about bioenergetics. All right, so once again, metabolism refers to all of the chemical reactions. And a metabolic pathway, which is what almost all metabolic activity in you consists of, is a series of small steps that complete a large grand process. So for instance, photosynthesis, as it says right here, we take carbon dioxide and water and ultimately produce glucose. Now, we don't just take CO2 and water and instantly make glucose in this process. There's a whole bunch of steps along the way. But in the end, by a multi-step process pathway, we do get to glucose in the end right there. Similarly, there's a metabolic pathway that extracts the energy from glucose, which is what plants themselves do. They make this molecule to store energy and then use it in distant locations. And of course, all animals are taking advantage of this stored metabolic energy in plants, glucose, and all the other things that a plant is made out of that was made using the energy harnessed in glucose. That material that the animals eat that was made by plants is broken apart into cellular respiration metabolic pathways, and in a long series of steps, ultimately we break that glucose back down into carbon dioxide and water. All right, a reminder that metabolism comes in two general forms. Anabolic metabolism, where we take small precursor molecules or even inorganic elements to start with, carbon dioxide, which is a molecule with inorganic material, and water, same thing. And with the source of energy, we combine those together to make larger, more complex molecules. Or, as this could be, 4 monomers of, say, amino acids or 4 monomers of carbohydrates, plus a source of energy, ATP in this particular case, used to power the synthesis of a polypeptide or a complex carbohydrate right here, which is a polymer of some sort. Catabolic metabolism, on the other hand, it takes large complex structures and breaks them apart, typically releasing energy. This could be a polymer being broken down into monomers, or this could be an organic molecule being broken down into its inorganic components right here. In either case, energy will be released. Anabolic, energy-consuming building reactions, catabolic, energy-releasing decomposition reactions. One of the reasons we study metabolism is to see how common the metabolic pathways are, or at least components of those metabolic pathways are in all living organisms. So here we've got a low-resolution tree of life with the 3 great domains, the eubacteria, the archaebacteria, and the eukaryota. And the ancestor of these organisms had a metabolic set of metabolic pathways. That's how it anabolically made the macromolecules that it's composed of, and catabolically extracted energy from things that it encountered to power all of that anabolic metabolism. And those pathways have been modified in current living eubacterial organisms. There's a hypothesis about that, but the diversity of metabolic pathways, but the shared commonality of features in all of those eubacterial metabolic pathways strongly suggests that they're all derived from a common ancestral system. Same thing for the archaebacteria, same thing for the eukaryotic organisms, including single-celled eukaryotes and the 3 great multicellular groups, plants, animals, and fungi. So just an illustration of how polymerization and depolymerization are examples of anabolic and catabolic metabolism. Here we're taking 2 monomers of glucose, combining them together to make a disaccharide. That's an anabolic metabolic path. Here we're taking a disaccharide like lactose or maltose or sucrose, breaking it apart into 2 carbohydrate monomers right here, and that's an example of catabolic metabolism. So think about the answer to this right here. And once you've made a decision there, why don't you cite what you used to generate that answer. Right. This should be reviewed based on previous schooling, but you should know that energy can be classified. All forms of energy can be classified as potential. That's the equivalent of stored energy. So fuel sitting in a fuel tank is potential energy. The charge in a battery is potential energy. Water behind a dam is potential energy. Stored, triglycerides in adipose tissue in an animal's body is potential energy, etc., etc. And then kinetic energy is the energy that is being released as things move. So water falling, is a kinetic energy situation. A battery powering a device, the flow of electrons through that device is an example of kinetic energy, and so on and so forth. And living organisms, just like forces in nature, can convert potential to kinetic and kinetic to potential energy back and forth. A very important type of potential energy and kinetic energy in living cells is the use of electrochemical gradients, as we'll see in cellular respiration and photosynthesis. If we can generate a large concentration of some particular atom or molecule in one location, and then allow it to go down its concentration gradient from one side of a barrier to the other side of a barrier, we can do work in that same process. So here might be, a huge amount of stored material right here. If it's allowed to move down its concentration gradient, work can be done at the same time. So that's an example of kinetic energy that can be harvested. The potential energy stored right here converted into kinetic energy as the material moves. I've talked to you about this before. So this is a hydrocarbon right here, carbons, hydrogens right here. An example of, one of the components of petroleum or liquid fuels, gasoline, and all of its relatives right here. And so that fuel, that chemical, stores potential energy. In living organisms, every organic molecule contains some amount of stored potential energy, particularly between the carbons and hydrogens. And you can see this molecule is all carbon and hydrogen bonds, so it has a very high energy density. Glucose has lots of carbon-hydrogen bonds, so that has a bunch of energy in it, and that's why glucose is an energy-rich molecule. A key bit of currency we want to be conversant in is this idea of Gibbs free energy. You're not going to need to worry about the heat of enthalpy or this formula right here and manipulate it. But G represents the potential energy stored in a molecule. Delta just means the change. So delta G is the change in the energy stored in chemicals as we go through a chemical reaction. All right, Gibbs free energy is a measure of the useful energy available in a molecule. So the question would be, if you compare glucose and water, which molecule has more potential chemical energy? In other words, that can be released by breaking bonds in that molecule, glucose or water. One of those two is going to have a higher Gibbs free energy, the other is going to have a lower Gibbs free energy. All right. So here's an example of an energy diagram, and in all of these diagrams, unless otherwise indicated, we assume time moves from the left to the right. So time is moving in this direction. So initially we've got a reactant or reactants right here, many copies of the same molecule or perhaps many different molecules, but let's just say a molecule right here with a certain amount of free energy stored in it that can be released. Now that reaction requires an activation. There must be an activation energy, kind of like lighting a fire to get a fire burning. This is the activation energy, would be the equivalent of striking that match, scraping the match on a abrasive surface to generate the energy required to light the match on fire. Or the match itself provides the activation energy to start burning some paper. That's this peak right here. And then the stuff starts burning, or in the case of metabolism inside a cell, this energy-rich molecule is being broken apart in a series of reactions, and energy is released. This is the Gibbs free energy change, the delta G right here, and we go from a large value, 10 units of energy, down to a low value, 1 unit of energy. So delta G would be -9 right there. The change would be in the negative direction. Energy is released. And the products right here would have less energy. All right, so this is an example of a catabolic reaction right here, going from a high initial stored energy to a low final stored energy. It can also be referred to as exergonic or sometimes as exothermic, because typically this reaction is going to release this energy. Some of it is going to be measurable as heat. That would be exothermic. Exergonic means it releases energy. This, on the other hand, is a endergonic, energy-consuming, heat-consuming, so endothermic, and anabolic reaction, where we take low-energy reactants with a provision of energy that has to be pumped in, plus enough energy to overcome the activation energy barrier here. So quite a bit of energy needs to be pumped in. We can convert these reactants to these final products. This would be a great example of what's happening in photosynthesis. We take water and CO2, the energy from a bunch of photons that's stored in various ways, but that's the ultimate source of that energy, sunlight. And we can break apart the components of water and oxygen into water and carbon dioxide into carbon, hydrogen, and oxygen atoms up at this high energy level right here, and then control the reformation into an energy-rich molecule, glucose, right here, that stores that energy, high energy level. Time, of course, was proceeding from the left to the right. So we've got two reactions right here. Which one is exergonic, giving off energy? Combination of these two monomers to make a dimer, or the breaking apart of this dimer to make two monomers? All right, activation energy. Activation energy is the energy required to get that chemical reaction occurring. So here I've got paper. It has a huge amount of potential energy in it, but it doesn't decompose and break apart into water, CO2 with a flash of light and all the other stuff that we can smoke and whatnot. Automatically, we have to provide an activation energy, which is typically the flame, lighting the paper on fire, or concentrated sunlight lighting paper on fire, or whatever you want. So in metabolism, a key role in all metabolism inside living cells, metabolism, is that we use catalysts, organic catalysts, which are enzymes, to reduce that activation energy. What that does is it makes this reaction much more likely to occur, and the energetics a little milder. We don't have to create such an extreme environment to get that chemical reaction to occur. So enzymes, which are an organic version of a catalyst just means some element or chemical that makes a chemical reaction occur. A catalyst, an enzyme, is an organic version of that, made out of protein almost inevitably. And what that does is makes the energy required to disassemble the components of our reactants much, much lower, so that it's more likely that they will break apart and then reform into their products. So here's an interesting idea right here. The breakdown of gasoline is another example of an exergonic reaction, and then it goes on to say that a spark is required to provide sufficient heat to exceed the activation energy. But once the reaction begins, enough heat is released to drive additional reactions. So yeah, to take some gasoline and light it on fire requires a spark. That's this activation energy right here. But then, if we measure this distance right here is greater than this distance right here. So the energy released by gasoline burning is more than enough energy to overcome the activation energy of other unburnt gasoline and convert it into the transition state where it can burn. And so the burning gasoline becomes a self-reinforcing process. Think of this as happening to one molecule, and the energy released right here is enough to disaggregate the components of another molecule of gasoline all the way up to this activation energy, and it can then release all that energy, and so on and so forth, for the reaction to be self-sustaining, self-feeding. The key thing is this distance right here, this change in energy here, must be greater than, or at least equal to, but really greater than, this energy level right here, the activation energy required to start the reaction. If this peak were greater than this distance right here, the chemical reaction would peter out very quickly. It would come to a stop. Right? Make sure that you can explain these two laws of thermodynamics. The first is that we can convert energy from one form to another, but we can't make it and we can't destroy it. So we don't want to say make energy or destroy energy. It's just convert energy. So we can convert energy, but we can neither make it nor destroy it. There's a fixed amount in the universe, which is a very interesting idea. The total energy content of the universe is understood to be fixed, but there are places where the energy is concentrated and where the energy is not concentrated, and those concentrated sources of energy can be allowed to diffuse over time and do work. The second law says that when we transfer energy from one form to another, from kinetic to potential, or from potential to kinetic, or back and forth multiple times between those two, we always lose some of that energy in the form of heat. That is irretrievable, lost heat energy. Second law of thermodynamics. In lecture I talked to you all about bouncing balls, and a bouncing ball goes from potential energy, the high position, it releases a whole bunch of energy as kinetic energy as it falls. When it hits the ground, it bounces back up. Potential energy of compression was converted into a rebound back up, but it won't go as high as it was initially. It will go some distance back up, bounce, come up some fraction of that distance, bounce, come up, bounce, come up, bounce, 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 until it's finally stopped. The loss of height at each bounce is an example of the second law of thermodynamics at work. So every step in chemical reactions, there's some energy lost as heat. ATP. What is its role in metabolism? So here, in this particular case right here, we've got these reactants, and we have to pump in a whole bunch of energy to get them up to their transitional state. And if we can secure them in a more complex form up here, like turning water and CO2 into sugar in photosynthesis, or taking single amino acids and combining them together to make a polypeptide, a polypeptide chain, this state right here, the energy must come from somewhere, and we know that that energy typically comes from ATP, and that's the role in metabolism. ATP provides a link between energy-releasing reactions and energy-consuming reactions. Very typically, it's going to be the hydrolysis of ATP, the breaking apart of ATP, that transfers that energy to get these reactants to their unstable condition right here, where they can be reorganized into something with a greater amount of stored energy. So ATP, adenosine triphosphate, here's the molecule right here. It has a purine nitrogenous base right here, adenine, the ribose sugar, that is typical of nucleic acids, and then 3 phosphates in a row right here. The gamma is the 3rd position, beta the 2nd position, alpha the 1st position. And what happens when ATP is hydrolyzed, normally that 3rd phosphate is broken free, transferred to the enzyme that is catalyzing whatever particular reaction that requires energy. And the transfer of the phosphate is the transfer of potential energy from one molecule to another. So for the record, the change in energy from the hydrolysis of ATP is about 0.73 kilocalories. It's negative because we go from a higher energy state, ATP, to a lower energy state, ADP, adenosine diphosphate, plus that one free phosphate now. So if I drew a reaction diagram for this, it would look like this. It would climb up, and it would drop down, and that would be ADP at the end of the we'd start at ATP up here, activation energy, and then drop to ADP and phosphate. And the difference between the initial state and the final state is 0.73 kilocalories per mole. So an example of something that does work using the power of ATP is that sodium-potassium pump that we looked at previously. So this generates a potential by accumulating lots of sodium outside of cells right here, making a very strong positive charge on the outside relative to the negative internal charge. Potassium is pumped in as well, but not as much potassium in as sodium out. That's where we get the charge difference. And we can pump both of these against their concentration gradients because we're using the energy of ATP to power the reaction. So again, what's the function of an enzyme? Reduce activation energy. Reaction with a catalyst. A catalyst in biology is going to be an enzyme of some sort. So we've talked about this quite a bit. All enzymes have reactants, also referred to as substrates, the things they act upon, that are specific to that enzyme. There may be several that it can bind to, but they're all shape related. And the idea here is that this protein has complex three-dimensional shapes on its surface or in its structure, and there may be many that have, that can potentially be the mirror or the inverse charge-wise and shape-wise of a particular substrate. So this thing is shaped like this, and it may have negative charges all over it. Well, you can bet the active site on this enzyme right here is going to have a similar shape and have negative charges all over it. If this has got positive charges, this will be all negative. If this is all negative, this will be positive. If this is negative, positive, 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 negative, this will be positive, negative, 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 positive. Something. There'll be an inverse mirror-like relationship between the two. Same for any other substrate. And when the substrates bind to the enzyme, if the reaction is exergonic or exothermic, the energy released in that reaction is often enough to drive the enzyme through its shape changes that catalyze that reaction. So in this case, the products breaking apart provides the energy to cause this to convert from its, this active state right here, breaking that substrate apart, back to its original state, where it can bind to fresh substrates and repeat the process. If we have, let's see if I've got a drawing right here. No, I don't. If we had a reaction, let's turn the arrows around in the other direction right here, and we had a endergonic endothermic anabolic reaction where we take two substrates, bind them to active sites on the molecule right here, join them together, and then release them as a complex substrate right here. We'd have to have an input of energy, and classically that would be ATP. So somewhere in here in ATP, at least one would be used if this entire process were running in the other direction. Here we're looking at a catabolic reaction. If I turn those arrows around, it would be an anabolic reaction. So typically of proteins, they're kind of soft and squishy, so the matching of substrate to active sites is not a matter of perfect puzzle pieces locking together. It's more like hands clasping each other during a handshake, and that's called induced fit. And so the substrate induces a change in shape of the protein. The protein grabs a hold of it, firmly right here, the enzymatic protein, and then does something, catalyzes some particular, reaction, whatever it may be. And again, if we're going to take small things and combine them together into a larger complex thing, we'll need an input of energy as well to make that happen. Yet the idea is induced fit. Think of hands shaking each other, or a hand grasping an object and kind of conforming to the shape of that object. And that's a little bit more accurate way of looking at the active side of a protein. So just as a reminder, and I've talked to you all about this quite a lot, all proteins, especially, active proteins, proteins that do things, as opposed to structural proteins like collagen or keratin, but active proteins that do things have a three-dimensional shape that can alternate between at least two different states, if not more. And that three-dimensional shape is a result of the particular unique sequence of amino acids in the polypeptide chain, and whether we've got non-polar, polar, negatively charged, positively charged, where are we? Positively charged, negatively charged, etc., amino acids is responsible. That unique sequence of amino acids is responsible for the 3D shape of the protein. It's also why it's very important if we have a mutation in DNA that changes the DNA code, we end up calling for a different amino acid sometimes in a particular location. Alter an amino acid from, say, a negatively charged one to a non-polar one, and we may dramatically alter the three-dimensional shape of the protein and change its function. That three-dimensional shape is extremely sensitive to environment. So classically, temperatures and pH, and to a lesser degree salinity, can all alter the shape of a protein as we change those values. Higher temperatures, we change the shape of a protein. Cool it down, we change the shape. Same thing for pH or salinity. So what does an enzyme really do? It provides stress, as it says right here, so that the substrates are less chemically stable and more likely to react. They're in a position where we can reorganize atoms more easily on it. They make it more likely that two atoms will interact with each other forcefully enough to alter their electron valence structures and create a chemical bond. Let's go ahead and take a look at this animation here real quick.
Every single second, inside every living cell, thousands of chemical reactions are taking place. These reactions are performed by enzymes. An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which is a series of chemical reactions used by cells to generate energy from carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. Containing power of enzymes lies in the fact that they catalyze reactions in mild conditions of pH, temperature, and atmospheric pressure. The rates of catalyzed reactions are millions to trillions times faster than those of the same reactions uncatalyzed. To speed up a reaction in the absence of enzyme, additional energy would need to be provided as heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier for the reaction to occur.
The result of the reaction
To
occurs
understand
more
how
rapidly.
enzymes work, let's take a closer look at a reaction in the citric acid cycle that is catalyzed by the enzyme aconitase. Aconitase binds to its substrate citrate and removes a hydroxyl group and a hydrogen atom to form the intermediate cis-aconitate. It then adds the hydrogen and hydroxyl back in slightly different positions to form the product isocitrate. In the active site, some amino acids are perfectly positioned to recognize the substrate and hold it in the optimal position for the reaction to begin. Some amino acids are involved in recognizing and holding the substrate. Other amino acids are directly involved in catalysis. Histidine 101 acts as an acid by donating its proton. Thanks to the chemical environment of serine 642, it can act as a base by accepting the proton from the substrate. The active site of aconitase also contains an iron-sulfur cluster that stabilizes the substrate electrostatically and helps to position it relative to the catalytic residues. The first step in the reaction is dehydration. In this step, histidine acts as an acid and protonates the hydroxyl on the substrate, allowing it to leave as a water molecule. Serine then acts as a base, extracting a hydrogen atom from the opposite side of the substrate, forming the intermediate cis-aconitate. Cis-aconitate then flips upside down, and the complementary hydration reaction is performed. In this step, histidine grabs a hydrogen atom from a passing water molecule, placing the resulting hydroxyl group back onto the substrate. Serine then returns its hydrogen atom, and the final product isocitrate is released. Notice that the enzyme itself was not changed by the reaction. It extracted a hydroxyl group and a hydrogen atom, and then put them back, thus starting and ending in the same state. This is the hallmark of a catalyst. When it finishes a reaction, it is ready for the next, so it can perform thousands of reactions in a row. Notice also that the shape of an active site is often flexible. Many enzymes surround their substrates, enclosing them to form the perfect environment for a reaction. Enzymes are fundamental to life on Earth, working every second of every day to maintain life processes in every cell, in every living creature on the planet.
All right, I hope you found that illuminating as to the actual mechanics of enzymatic reactions or enzymatically catalyzed reactions inside living organisms. That particular reaction is from the cellular respiration process. You don't need to memorize that, but you do need to be able to answer some questions about active sites, intergonic and exergonic reactions, which ones are going to need an external power source like ATP, and what happens to the enzyme in a reaction. Is it consumed or does it remain ultimately unchanged? All right, a little bit about regulation of enzymatic pathways. Regulation ensures that a metabolic pathway only occurs when it's needed at a rate that is commensurate with, the needs of the organism, so that materials are not wasted. And so we must have some sort of regulation. All right. So an important idea is the idea of competitive and non-competitive inhibition regulation of a enzyme. So the gray diagram right here represents some particular enzyme structure right here. and we've got a substrate. In this particular drawing right here, we've got a substrate and an inhibitor, and the substrate binds to an active site where conversion of that substrate occurs right here. This thing, let's say it's a disaccharide, binds to the enzyme. The enzyme cycles through its shapes right here and breaks the disaccharide into two monosaccharides. Say this is sucrose, and now it's glucose and fructose right here. Well, here's an inhibitor that is a non-competitive inhibitor. It binds to a different site on the enzyme, but causes it to not be able to take its active shape for converting the substrate right here. So the non-competitive inhibitor binding this other location right here doesn't compete with the substrate itself, but it makes it impossible for the enzyme to complete its cycle of shape conversions, and therefore it stops the enzyme from functioning. Now, thermal energy in this environment, as long as we have some thermal energy, all these molecules are vibrating and bouncing around and moving around. Sooner or later, that inhibitory molecule will be popped out of here, at least there's a probability, in which case this thing can cycle through one of its enzymatic reactions, but then there's a probability of another inhibitor binding to that location or the same one binding again. The higher the concentration of the non-competitive inhibitor, the more likely it is that you'll completely disable this chemical reaction. When we look at a graph of enzymatic reactions, and this is something that you will have on your test right here, I want you to be able to recreate this particular situation here. So what we're looking at is the concentration of substrate. There's nothing here. Say we're looking at this previous reaction right here where we're taking sucrose, a disaccharide from plants, and breaking it apart into fructose and glucose right here. So let's assume that that's the reaction. So here we have no sucrose at all, and here we have greater and greater concentrations of sucrose. And we've got one copy of the enzyme. So if there's no sucrose, the one copy of the enzyme can't do anything. But as we increase the concentration of sucrose, we have a higher and higher probability of a sucrose molecule binding to the active site of the enzyme and the enzyme catalyzing that reaction. And ultimately, so as the concentration of the substrate increases right here, the enzyme is able to catalyze more and more reactions until finally the enzyme is catalyzing that reaction as fast as it's physically capable of rotating back and forth between its initial state, its transition state, and its final state. And that's going to be the maximum rate of that reaction under those conditions. If I double the number of enzyme molecules, then of course I would, I would get a taller curve right here with more reactions occurring per unit of time. But let's just assume this is one enzyme. So if I have a competitive inhibitor right here, as I increase the concentration of the competitive inhibitor, something that binds to the same place that sucrose binds to, I'm going to slow the rate of reaction down because periodically the inhibitor is binding instead of the target substrate. And so, and I have a certain amount of that say I've got one copy of that competitive inhibitor binding to one copy of the enzyme. But as I increase the substrate, the sucrose right here, inevitably that inhibitor is going to pop off for a moment. And the higher the percentage of everything in that test tube, let's say, or that cell, the greater the concentration of substrate, the more likely it is that a substrate molecule will bind to the enzyme rather than the competitive inhibitor. And so eventually I get to such a high substrate concentration right here that I get to my maximum rate of reaction anyway, or danged close to it. These never quite join to each other, but they'll get ever closer and ever closer. On the other hand, a non-competitive inhibitor doesn't need to compete. That's why we call it non-competitive. So, one copy of it is, in this particular case right here, will permanently reduce the rate of the reaction right here. This height of reaction right here represents the frequency at which the non-competitive inhibitor breaks away from its active site and allows the enzyme to momentarily do its chemical reaction before the competitive inhibitor binds again. And the idea there is that all of these things bind and let go frequently. If they bound permanently, all these proteins would be instantly disabled. So a relationship between non-competitive inhibition and this term allosteric inhibition is a good thing to talk about. So allosteric inhibition just means that there is a molecule that will bind to a site other than the active site for the substrate in question on an enzyme, and when it binds there, it changes the shape of the protein so that it can no longer act on its substrate. That's really the same thing that we were looking at right here. With this thing bound here, the enzyme can take on its permanent shape and bind to the substrate correctly to catalyze its reaction. And this is a very typical regulatory process. So this enzyme may catalyze, this enzyme is going to be part, let's see if we've got a sequence, yeah, here we go. So this enzyme right here is a part of a reaction sequence. We've got a bunch of enzymes catalyzing various steps along this process right here, and the final product, if its concentration becomes high enough, it can allosterically, and often does allosterically, regulate the behavior of the first enzyme in a metabolic pathway. So like, this is a product amino acid in a metabolic process that's converting one amino acid into a completely different amino acid. And when we've got enough of that amino acid, the cell's not using it up rapidly, that's going to inhibit the conversion of this amino acid into that. That is a negative feedback regulatory loop, and the particular mechanism that's utilized is allosteric inhibition right here, where this molecule binds to a different active site, if you will, or a different site on the enzyme, changing its shape so that its active site for its target substrate is no longer compatible. All right, and that is the material for the review lecture on metabolism. Go back over this material along with your cellular respiration and photosynthesis materials, and that is the subject matter for the Unit 3 exam.